All right, so our last bit for the Hardy Weinberg Lab is figuring out how to display our data, because right now it's just a whole lot of numbers, so we need to get it into shape. So I'm going to share my screen with you. I'm going to share it to my Excel document. Okay, so the problem with this lab, or one of the main challenges that students have with this lab, is that there is different ways of different scientific questions require different data analysis displays. So I had a very specific hypothesis with my scientific question. My scientific question, what would happen if I killed off half of my homozygous recessive organisms? And so my hypothesis is that my Q is going to go down. And so that's fairly easy for me to show. And I'll talk about a different, some of you, okay, no, I'll do it right now. Some of you, on the other hand, might expect more variability. Let's say you did a bottleneck, right? Or let's say you're comparing population size and you did a population of 10 and compared it to a population of 100 and compared it to a population of 1,000, right? Then you would expect variability, and showing averages doesn't show us variability. So if you expect a P or a Q to go up and down, it's perfectly fine to show your averages. If you scientifically expected that, right? Not just, oh, I think this will happen. But if you expected variability, there's a very different way you're going to have to display your data. So I expected my Q to go down, so that's what I'm going to see. I'm going to look at right now. So I'm just going to go ahead and find my average. I'm going to find my average P. Parenthesis, using that formula equals average, highlight my data, enter. I'm going to do that for my Q. And I'm going to drag it all over. Okay, it doesn't like some of them. So the problem, okay, so here now I have my averages. Right now, if I'm just like, hey, Excel, graph this, it's going to have a really hard time because it's not organized in a very nice way. Yeah, so I'm going to have to take a minute to once again organize everything before I can, why I'm bothering to delete these, before I can go ahead and ask Excel to help me make a graph. So I'm going to arrange this into sort of a data table. So I'm going to Control C, and I'm going to paste special my numbers. Okay, and so this is generation 1. This is generation 1, and this is my P, and this is my Q. Okay, so now I'm going to make a table that has my generations. I think I had 10. So now we're like layers upon layers upon layers of organizing our data, right? It gets a little ridiculous feeling, but this is life. First generation, second generation, copy, paste special, numbers, copy, paste special, numbers. That was 4, here's 5. Numbers, 6, copy, special numbers, 7, copy. Special numbers. 8. And 9. And 10. Okay, so now I have all my data. Okay, and so now it's a lot easier to be like, hey, Excel, graph my data. So I'm just going to highlight my data. I have to go hide in my son's room to get a little quiet. Things are a little crazy around here today. I'm going to insert and I want an XY scatter plot with 2 lines on it. I think it actually worked out. Okay, so my blue is my P and my orange is my Q. And so I can go in, I can add some axes titles, right? And down here, this is my generation. And over here, this is my allele frequency. Okay, and my chart title is change in allele frequency after environment changes. The heroin MENT changes after generation 3. And so when I go to present this, I'm probably going to want to put a line on here or something like that to make that a little more obvious that something's happening in generation 3, but my graph makes it pretty obvious. If you remember how to add error bars from when we did that back in the beginning, I could also graph the standard error of the mean on these. I'm going to find my standard deviation, divide by the square root of my n, and add error bars so I could see if my variability increased or decreased. So if I had the kind of scientific question where I expected a P or Q to go up or down, I could basically stop here. I would talk about some limitations, but I can use this to answer my scientific question. Yes, as I kill off half of my homozygous recessive individuals, the allele frequency for Q goes down. I could talk about if this went on for 1 million generations, I wouldn't necessarily expect all my Qs to die out because there's no disadvantage of being heterozygous. I could talk about that limitations of this model is that my population chain always stays the same, that this is the only selective pressure, that there's no mutations, right? All these different things that aren't true in real life because it's model. And that's the way it is. Okay. Now, this is, so I'm going to make a copy of this spreadsheet, and I'm going to walk you through how I would analyze my data if I were expecting variability in my data. So this one gets a little tricky. I'm going to rename this. Rename. Okay, because I'm going to have to do some editing. So one good way of showing variability in your data is to look at what happens to sort of individuals. So I'm only going to focus on my P in this case. I could focus on the P or I could focus on the Q, but it's really easier just to focus on one of them. So I'm going to delete all of my data except for the P. And I can do this a little faster by highlighting these two. And again, no, not cut, delete. No, just delete. Okay. I'm doing this on a copy of my data, so just for in case I change my mind, I don't need to freak out because I have it all there. Delete. Q. Delete. 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 Okay, so to show variability in my data, I'm not going to use any averages. So I'm going to call these trials over here. Trials, and I have trials 1, 2, 3, 4, 5, 6, 7, 8, 9. I probably should have done this earlier. 11, Why am I typing these all by hand? I know how to work smarter. Drag it down, it'll automatically number for me. Okay. Let's see what happens if I just tell it to graph this. Oh, I'm going to remove all these P's because I don't want that on there. I want it to go by the trial. Oh, Excel did this for me nicely. What? Stop. Okay. Insert. I think I want a scatter plot. No, I think I want just a line graph. That's not what I wanted. I wonder if I can make this work though. I'm going to go to my select data. Because I want this series name is 1 and its values are this right here. Okay, and then add. And my series name is 2, and I want it to be these values here. So you see where I'm going here? The point of this one is that at the end of the day, I will have a graph with 26 lines on it because apparently I had 26 trials. I was going for 25. And each line will show one of my trials. So I'll really be able to see trends in my data, but I'll also be able to see variability. So if things start one way and they go kind of crazy all over the place, this is a really good way for me to be able to see this. And I'm not going to graph all of them because there's one more thing I want to talk about. But this is a way in which you can show variability. There's other ways too. Like with those averages, if I had gone back and made my error bars, that was another way I could show variability. So I'm going to stop here. I would expect you to keep going, but I don't want to waste your time adding a gazillion lines to my graph. So, but I do want to, of course, label it. I want axes titles. This one down here is generation, right? I had 10 generations. And this one right here is P. Frequency, right, of P allele. And I could make another graph of my Qs if I wanted to. My key is my individuals. We always wanted part title. And I would call this change in allele frequency over 10 generations with selective event beginning generation 3. Okay, so that's a way that I might title my graph. Okay, so the last thing I want to talk about is control. I had a bit of a control built into my graph here because I didn't do anything in the first 3 generations. It was just, that was where I had no selective pressure. I was hopefully within the confines of Hardy Weinberg in those first 3 generations. So that could be considered my control. Another way to have a control is that if I ran another 26 trials with no selective pressure, so another 26 trials where there was no, where I didn't kill off half of my QQs like I did, and then I could compare what I found to that. So either of those are acceptable controls. I kind of like having both of them in there, but we don't need to be too over the top at this point now, do we? So yeah, at the end of the day, that's how you graph your data. And let me know if you felt like that this is not a good representation of your data, and I would be happy to help you figure out some other way to do that. So other than that, I hope you are doing well, and I'll yeah, let me know how it goes.